In our last video, we took a close look at the structure and function of fatty acids. Fatty acids, of course, are one of the key components of our complex lipids. So we saw how our fatty acids can be linked into storage lipids when we combine them with glycerol, and that acts as one of the main energy storage sources in our body. In this lecture, we're going to be focused on the complex lipids that are involved in membranes. So all of these will involve fatty acids in one form or another, but will be linked to other molecules in order to create a variety of different membrane lipids. Our membrane lipids will all share two key features. So they'll have nonpolar tails, which will consist of our fatty acids normally, and then we'll have a more polar head group. So this will create an amphipathic molecule with both polar and nonpolar characteristics, which will be key for building our membrane structure. The diversity of our membrane lipids comes through several different means. So we can change up what backbone is used to connect our polar head groups and nonpolar tails together, right? So previously we've seen glycerol used to link our different fatty acids together in our triacylglycerides, but we'll see some different molecules that we can use to link our fatty acids and head groups together as well. We can also change what fatty acids we're using. So again, we saw that if you have longer fatty acids or unsaturated fatty acids, that would change up your melting point for those fatty acids, with longer fatty acids having higher melting points, and double bonds or unsaturated fatty acids causing lower melting points. We can also, of course, change up our head groups as well. So the properties of our head groups will be key for determining what properties our membrane will have. Right, whether we have negatively charged head groups that will then interact with their positively charged molecules, or whether we have hydrogen bond donors that might be participating in hydrogen bond formation. And we can then change up what head groups we have based on what we want the properties of our membrane to be. So we'll actually see key differences in our lipid head groups based on what our organism is, or what tissue, or what particular membrane we're looking at. The most common form of membrane lipid is our glycerophospholipids, also called phospholipids. These phospholipids consist of two fatty acids linked together using ester bonds to glycerol, but we're going to use a glycerol that also includes a phosphate here. So we have two alcohol groups used to link fatty acids, and a third alcohol group used to link to our phosphate. The phosphate, of course, will create a negative charge at physiological pH. So this is a key component of making that polar head group for our phospholipid. When we combine these features together, our phospholipids will look something like this. So again, we have our fatty acids here in yellow. These are linked to our glycerol backbone through those ester bonds. And then we have our phosphate linked on to the third alcohol of our glycerol. And we can also attach additional head groups to that phosphate through a phosphodiester to create a wide variety of different polar head groups. What we commonly see with most of our natural phospholipids is that we'll have unsaturated fatty acids at the second carbon of our glycerol here. So notice how we have a polyunsaturated fatty acid here with two different double bonds. And then we'll tend to have a saturated fatty acid at the first carbon of our glycerol. So carbon 1 is saturated, carbon 2 with our unsaturated fatty acid attached, and then carbon 3, of course, has our phosphate attached. Here are some examples of some of the common head groups that we'll see with our phospholipids. So we can range from things as simple as just adding a hydrogen onto our phosphate. So it's just a phosphate group essentially as our head group. Or we can add on even different amino acids such as serine, or complex sugar-like molecules such as this inositol ring structure. We can even attach two phospholipids together using this phosphate and glycerol linkage to create a very large kind of double lipid structure. The most common phospholipid in our eukaryotic cell membranes is phosphatidylcholine. So the phosphatidylcholine name comes from our head group here with our choline. So we have a positive charge with this quaternary alkyl ammonium and a negative charge from our phosphodiester. So this forms a Zwitter ionic lipid that will be net neutral, but will have a highly polar nature. While phosphatidylcholine is very common in our eukaryotic or mammalian cell membranes, it's very rare in bacterial membranes. So we'll tend to see some of our other head group varieties there as they don't contain the enzymes required to synthesize phosphocholine. For naming our phospholipids, so we're going to see a combination of our head group name, so that's our phosphocholine here, along with the names of our individual fatty acids. Because again, remember, we can vary up what fatty acids we have attached to our glycerol backbone here. So in this case, based on the length of our carbon chains, the number of double bonds, we could name these fatty acids. So they are stearic acid, which is a 18-0 saturated fatty acid, and linoleic acid, a 18-2 unsaturated fatty acid. Of course, we can further add in the positions of our double bonds here. So this would be a delta 9 and 12 for the two positions of our double bonds there. So again, stearic and linoleic are just the common names for these fatty acids. We combine that with our head group name of phosphocholine, and this becomes 1 steroyl, referring to our stearic acid chain at the first carbon, 2 linoleoyl, again referring to our linoleic acid branch at the second carbon of our glycerol here, SN glycerol 3 phosphatidylcholine. So again, the 3, of course, indicating the phosphate and our choline group attached at that third position. Now, of course, this name is rather long and difficult to say, so we abbreviate this as SLPC. So the S and the L, of course, referring to our fatty acids, and the PC is an abbreviation for our head group, meaning phosphatidylcholine. So you'll commonly see different varieties then of phosphatidylcholine as, for example, you might see POPC is a very common lipid in our body. So this would be palmitic acid, oleic acid combined with our phosphatidylcholine. So the same head group, just with different fatty acids attached. You'll notice here that we do tend to use the common names of our fatty acids, but then abbreviated within that 4-letter code for a fatty acid. I don't expect you to memorize all these fatty acids, but there will be some fatty acids that you'll tend to see very commonly, so you might want to familiarize yourself with those. So we have palmitic acid and stearic acid as quite common ones, and then of course our unsaturated fatty acids, we often see oleic acid or linoleic acid, and occasionally our arachidonic acid as well. So how do these phospholipids build up our cell membranes? Well, of course, as we saw, the key features of our phospholipids is that we have a highly polar head group and we have our nonpolar lipid tails. So when we place them in aqueous solution, as we know from our hydrophobic effect, our nonpolar tails will want to try to hide from our aqueous solution. Rather than forming a micelle, we tend to form a bilayer structure where if we put our lipid tails together and face our polar head groups outside, we can, again, hide all of those nonpolar groups from our water. Right? So our blue beads are representing those polar phosphate-based head groups. And of course, the yellow is the fatty acids forming that nonpolar group. The key really for forming this sort of bilayer structure as opposed to the micelles that we've seen previously is that we have a cylindrical structure overall for our phospholipid here, right? So the size of our fatty acids are a bit bigger since we have two of them attached to our single phosphate group. So we can't bend it around as easily as we can with the micelle, right? So in a micelle, you just have one fatty acid and your polar head group. And so it's easy to kind of crunch our fatty acids closely together as we bend them around into the spherical micelle shape. So our bilayer is better for these larger and more complex phospholipids, as well as, of course, our other complex lipids like the sphingolipids that we'll be looking at later. So as we've seen, phosphatidylcholine with its zwitterionic and neutral head group will act as the base for our membrane. We'll then add a variety of different other head groups in order to allow our membrane to interact with other biomolecules. So depending on what head group we add, we can have anywhere from 0 to as high as around -4 for the charge of our phospholipid head group. You can see they always tend to have negative charges, which of course then we can use to interact with positively charged proteins. So by changing the amount of negatively charged lipids or what negatively charged lipids we have, we can change up what proteins will interact with. You can also, of course, note the presence of other hydrogen bond-owning groups, for example, with our phosphatidylglycerol or phosphatidylethanolamine. So that will, of course, add to the complexity and range of different interactions that we can make with our binding partners. Since the interactions of our membrane will mostly be determined by what phospholipid head groups we have, we'll tend to see a wide range of different phospholipid compositions between different membranes. So this will allow these membranes to interact with different kinds of proteins or different biomolecules. So you'll notice this chart here is showing kind of the range of different kinds of lipids in each of these different membranes. We have our plasma cell membrane, but then, of course, we have membranes around each of our organelles. And this chart here, of course, is showing composition of all lipids beyond just our phospholipids. So you see phosphatidylcholine, of course, is a major component of all of these membranes. We also see a lot of phosphatidylethanolamine. The minor lipids shown in yellow will be a variety of those different negatively charged lipids. And then we also have cholesterol and sphingolipids, which, of course, we haven't looked at their structures yet, but they do make key components of our membranes as well. So as that last graph showed, we see a large variety in the amounts of different lipids within each of our organellar membranes. The other thing that we see is we see different kinds of negatively charged lipids found on different membranes. So this can act as essentially a recognition signal for our membranes, where any proteins that are interacting with our membranes can see specific negatively charged lipids and use that to either bind to the correct organelle based on what lipids are there. So we can specifically see this comparing our endoplasmic reticulum versus the cytosol. So notice how we have these yellow phosphatidylinositol phosphate lipids here in the plasma membrane, where we have more of our purple phosphatidylserine lipids and our pink phosphatidylinositol lipids without any of our phosphatidylinositate phosphate yellow lipids in the endoplasmic reticulum membrane. So just by changing what lipids there are, again, it will allow proteins to recognize what organelle they might be interacting with. Another thing that you might notice here is that we actually have a very different lipid composition between the two faces or leaflets of our bilayer. Right? So our bilayer has these two rows of lipids, or these two layers of lipids essentially stacked on top of each other to create that nonpolar core. And the inner layer, or the inner leaflet of our lipids, looks quite different from the outer layer, right? So we have a lot of different kinds of lipids on the inside, whereas on the outside, it's mostly just these green sphingolipids and blue are phosphatidylcholine. Compare that to all of the phosphatidylinositols and phosphatidylserine as well as phosphatidylethanolamine that we see on the inner leaflet. So our membrane is actually different in composition, or asymmetric, we would say, across the two leaflets of our membrane, as well as, of course, between different organelles. So in this chart, we see a quantitative measure of that membrane asymmetry. So on the left, we have our inner leaflet. On the right, we have our outer leaflet. So you'll notice for sphingomyelin, one of our sphingolipids, it's mostly on the outer leaflet with a little bit on the inner leaflet. Our phosphatidylcholine as well is primarily found in the outer leaflet, whereas on the inner leaflet we see phosphatidylethanolamine, and we also see phosphatidylserine, which is exclusively found on the inner leaflet. Phosphatidylserine, of course, is one of our negatively charged lipids with a charge of -1, and so we end up with an inner leaflet that is enriched in negatively charged lipids. So we'll have all this negative charge on the inside, whereas on the outside, these are all zwitterionic or net neutral lipids. So our membrane actually has a difference in charge between our two leaflets, in addition to that difference in our lipid composition. This membrane asymmetry is possible because the transverse diffusion of our lipids is relatively slow. When we refer to transverse diffusion, of course, this would be our lipids flip-flopping from one face of the membrane to the other. And as you might imagine, this process is relatively slow because we have to take this polar head group and push it through to the other side of the membrane. So for the most part, our lipids just kind of sit on one face of our membrane, and it's relatively rare for them to flip over. It does still occur, right? We will occasionally see lipids flip over, but it's a relatively slow process, right? So over a period of days, your lipids will kind of slowly flip back and forth, but it takes a long period of time. Because our lipids slowly flip-flop across the membrane, it's important that we have proteins that will control this sort of transverse diffusion, right? So if we want to be able to keep phosphatidylserine only on the inside of the membrane, we're going to have enzymes that will take our phospholipids and flip it to the inside when it flips to the outside. So we have enzymes we call flipases that take PE and PS that might flip by itself to the outside and flip it back to the inside to our cytosolic leaflet. We also will have flopases that will take other lipids and move them to the outer leaflet. So a flipase moves it in, and a flopase moves it out. So the flopase, of course, would be used for phosphatidylcholine or sphingomyelin in order to keep those more enriched on the outer leaflet, while we have our negatively charged lipids enriched on the inner leaflet. There are also some enzymes known as scramblases that will move lipids in either direction. So they'll just allow your lipids to mix until we have an equal amount of lipids, for each kind of lipid on each membrane leaflet. This membrane asymmetry that we build using these flipases and flopases has a very special significance. So normally, of course, phosphatidylserine is only on the inside of our membrane, and so that will allow proteins on the inside to bind to the negatively charged lipid. During apoptosis, our phosphatidylserine gets moved to the outside through the action of scramblases. So our apoptosis will activate scramblases, and so we'll end up mixing our phospholipids between the two leaflets. So you see our blue PS now on the outside, and red would be representing phosphatidylcholine is now on the inside. Of course, we always have some phosphatidylcholine on the inside, but now we'd have a higher concentration as we are equilibrating our phospholipids on either side of the membrane. This will then reveal our PS on the outside of our membrane, where it can bind to various proteins, including our annexin 5 which then will mark the cell for destruction. So this membrane asymmetry then enables us to send a signal that our cell needs to die and we need to move into apoptosis. In addition to our phospholipids, we also see sphingolipids play a key role in our membrane structure. Sphingolipids differ from our phospholipids in that they use sphingosine as their key backbone rather than glycerol. So our sphingosine here is rather unique in that it has a single amine group as well as two alcohols that are used to bond on our different head group and fatty acid. We only use a single fatty acid linked to that amine through an amide linkage, and our long carbon chain here of our sphingosine essentially acts as our second fatty acid to complete the typical kind of membrane lipid structure. For our head group, we're going to use either a glycosidic linkage or a phosphodiester linkage, right? So we could link on a phosphate here and use our typical phosphodiester-based head group linkage, but we can also use that alcohol group to make glycosidic bonds with sugar molecules. Here you can see some of the varieties of different sphingolipid head groups. So we do have phosphocholine as a possible head group here, just as with our phospholipids. We will commonly see sugars attached, so that would be through our glycosidic linkage to that alcohol. And we can have a variety of different complex oligosaccharides attached through that linkage here. So this will be some of those glycoconjugate glycolipids that we've seen previously. Our simplest sphingolipid is ceramide. So ceramide would just have a single hydrogen attached to that sphingosine backbone, and so it would be mostly relying on the polarity of the sphingosine head group region with the alcohols and the amine group to act as our polar head group for that sphingolipid. Our most common sphingolipid is sphingomyelin. So this would be if we take ceramide, or our sphingosine plus the fatty acid, and add on phosphocholine. So sphingomyelin is a major component of our cell plasma membrane. It's also very abundant in the myelin sheath that surrounds nerve cells in animals. So that myelin sheath is critical for increasing the speed of signal transmission through our nerve cells. You'll notice, of course, that sphingomyelin is very similar in structure to our phosphatidylcholine. They both use the same phosphocholine head group. We just have, of course, that key difference in what backbone molecule we're using, whether it's glycerol or our sphingosine. Where our sphingolipids are most unique is in their glycosphingolipids. So again, of course, we've seen all of the different symbols that we can use to represent our different kinds of sugars. So as you can see, we can get quite complex oligosaccharides attached through, again, combinations of various sugars here, right? So we have glucose combined with galactose, and our modified sugars as well, the N-acetyl-galactosamine, as well as our N-acetyl-neuramidic acid. And of course, we can continue to extend that to make more complex glycolipids as we build up that sugar polymer. Now, of course, we previously saw these glycosphingolipids when we looked at glycoconjugates and our blood types. So we saw the structure of our O antigen, our A and our B antigens previously, and we talked about how there's a difference in the sugars attached at the end of these glycoconjugates. And of course, now as we take a closer look, you can see that this uses our ceramide as the backbone that's then linked onto our sugar chain here. So we have our typical ceramide with the sphingosine and our fatty acid, and then we have that glycosidic linkage to our sugar chain here to complete our glycosphingolipid. In addition to acting as the key antigens that determine blood types, glycosphingolipids also mediate a large range of other interactions, particularly between cells and other cells or extracellular molecules. Many of these interactions are mediated through these ganglioside glycosphingolipids. So our ganglioside lipids include, of course, our sugar oligosaccharide attached onto ceramide, but they also include this sialic acid sugar here marked in purple. There are a wide range of different kinds of gangliosides that each will have unique roles depending on their exact structure of that sugar chain there. Because of their importance in a wide range of different interactions, dysregulation of the gangliosides can lead to a wide range of different diseases. So in this figure here, we see the recycling pathway for a variety of different gangliosides. So in each case, we have different enzymes that are involved in removing sugars to convert between these different ganglioside structures and eventually free up those gangliosides and just release our ceramide. For each of these enzymes, if we have an error in that enzyme, it will lead to a variety of different diseases. So you can note all of these different diseases here that are caused just by errors in the enzymes that are needed to remove these sugars and recycle our gangliosides. As we see with our gangliosides, the recycling of our lipids is very important for controlling our cell behavior. So we'll often need to recycle different lipids in order to change the interactions at our membrane surface with other biomolecules. In the case of our phospholipids, we're going to have a different set of enzymes that we will target to recycle our phospholipids. So we call these enzymes lipases, and our lipases will be targeting the various ester bonds in our phospholipid. So of course, we can target the ester bonds linking our fatty acids onto our glycerol backbone, and we can also target the phosphoester bonds that connect our head group to our glycerol backbone. Depending on what lipases we have, we'll actually change which ester bonds we target. So we can specifically recycle different portions of our lipid. So you could cut off just the head group and then replace it with a different head group, for example, to change the interactions that would occur then between that lipid and other proteins, for example. So we'll have different lipases that are targeted towards our different ester bonds here in our phospholipids. So the phospholipase A1 will target the fatty acid connected to the first carbon of our glycerol. The phospholipase A2 will target the fatty acid on our second bond here, our second carbon rather, of our glycerol. And then we have our phospholipase C and phospholipase D that will tend to target on either side of our phosphodiester. So C will cut off the entire head group, including our phosphate, whereas D just cuts off the unique portion of our head group and leaves the phosphate behind. So the identity of the phospholipase will determine which of our ester bonds is cleaved. This phospholipase degradation has a couple of different goals. First off, again, it allows us to recycle our lipids, right? Whether we want to replace our fatty acids to change up our lipid composition slightly, or replace what head groups we have, it allows us to kind of vary the phospholipid composition of our membranes. The second goal is that will allow us to release lipid molecules, whether it's cutting off a fatty acid or cutting off a head group, and these can actually react and create what we call biologically active lipids. So biologically active lipids are lipids that take an active role in signaling within the cells. So there's a wide range of different kinds of biologically active lipids. And of course, there we see a very different role for our lipids compared to the more storage or structural roles that we've seen so far. So we're going to take a closer look at the different kinds of biologically active lipids in our next lecture.